Apoptosis. Apoptosis signaling. Apoptosis, or programmed cell death, is a rapid self-destruction process that results from the simultaneous activation of lipases, proteases, and nucleases, called caspases collectively, that fragment cell structures. During apoptosis, destroyed cell components are randomly packaged into small vesicles that are phagocytosed by macrophages, leaving no trace of the dead cell. Apoptosis can be initiated from inside or outside the cell via any one of several intersecting signaling pathways. The cell death program is a default signaling pathway, always available if a cell becomes damaged, infected, or redundant. Apoptosis was first described in nematode worms, Caenorhabditis elegans, and has become a central topic in cancer research and immunology. Healthy cells receive a balance of pro-apoptosis signals and anti-apoptosis signals at any given time, stimulating intersecting signal transduction pathways. If the balance shifts toward apoptosis, the pathway's off switches are deactivated and caspases fragment the cell in about 1 hour. It may seem counterintuitive to have a death signal program ready to run at all times in healthy cells, but in multicellular organisms, precancerous, infected, or very old cells can do more harm than good. During animal development, apoptosis shapes tissues as well. This tadpole will lose its tail as it develops as a result of apoptosis. BCL2. BCL2 was the first human cytoplasmic protein discovered that is involved in regulating apoptosis. It is encoded by the BCL2 gene. 24 additional members of the BCL2 family of genes have been discovered, and all are involved in regulating apoptosis. BCL2 protein has been shown to interact with 30 other signal transduction proteins, which is evidence for the complex interplay of signaling pathways. BCL2 was discovered in a B-cell lymphoma, a cancer of white blood cells that had a mutated BCL2 gene. BCL2's normal function is to inhibit apoptosis in healthy cells. When the gene is mutated in cancer cells, the protein is always active, always preventing apoptosis, even in cancer cells that should have died to protect the organism. Inhibition of apoptosis pathways is one requirement for the development of a tumor, and many tumors have a mutation in the BCL2 family of genes. Apoptosis may be initiated from signals inside the cell, like the nucleus and cytoplasm, or outside the cell through the membrane. From the nucleus, failure to repair damaged DNA sends a signal to BCL2 family proteins in the cytoplasm via P53, a master regulator protein of the cell cycle and DNA repair enzymes. P53 activates pro-apoptosis enzymes and inhibits off switches such as BCL2. This is the major pathway that protects us from cancer. From the cytoplasm, abnormalities in metabolism or in signal transduction pathways initiate apoptosis via BCL2 family proteins. Activating apoptosis via this pathway prevents tissue damage from cells that are producing toxic byproducts. From the cell membrane, white blood cells release ligands called death factors that bind to the membrane receptor FAS. FAS signals activate caspases directly without the involvement of the BCL2 family proteins. This pathway regulates the healthy development of white blood cells and also leads to the death of virus-infected cells and cancerous cells. The fast ligands or death factors may be autocrine, juxtacrine, or paracrine signals depending on the context. Apoptosis signaling is connected to many aspects of cell signaling and responses and can be initiated from inside or outside the cell.